And I think we got up very close to the end here. We were speaking about the male regulation of hormones and so on. And then we got up to speaking about the development of sperm. So we'll finish this up and then we'll move into packet 2, which is the female reproductive system. So someone is asking about weekly quizzes. Yes, I am being very flexible. If you missed a weekly quiz, they all should be available. If you missed any of those, please go ahead and get caught up. And if it's not available, if it's one of the ones that was from previous units before all of the madness that happened after spring break, let me know as well. I can open those up so that you can complete those. So yeah, definitely reach out if you missed. The lab quizzes, only the quizzes that are available on Blackboard, I'll be waiving. So if there's a quiz that's available on Blackboard, you should still have access to complete it, but the ones that were before spring break, you will not be able to make up, obviously, okay? All right, perfect. So we left off here looking at sperm development, and let's talk a little bit about what a mature sperm looks like, and then we'll speak about how sperm develop. So we've got 3 parts to a mature sperm. There's the aquasome, there's the head rather, which has the aquasome and the nucleus. There's the mid-piece, sometimes referred to as the neck, and then there's the tail, which provides the motility for a sperm. So in the head, we've got the chromosomal area, which is in the nucleus. This has the DNA and all of that information, which will provide fertilization to an oocyte. But we've also got the aquasome, which has the enzymes that will actually burrow into the layer of the egg. So we'll talk about the zona pellucida, which is a thick membrane that surrounds a mature oocyte. And in order to actually burrow through that zona pellucida, there needs to be enzymes to degrade that layer, and they are present in the aquasome of the head of the sperm. The midpiece has the mitochondria. Obviously, that's going to be needed to provide energy, to actually energize the sperm, to allow it to move, give it some motility. And then the tail has this whip-like portion, which actually propels or provides easy movement, which we call kind of swimming throughout the reproductive structures of the male as well as the female reproductive path. So this is what a mature sperm looks like, and if everything is working, it should have a well-developed tail, it should have adequate mitochondria, and then enzymes for actually burrowing through the egg. Let's look at how they develop. So this is a cross-section of the seminiferous tubule. We talked about seminiferous tubules last class, and this is what a cross-section of that tubule looks like. So they are lined by the Sertoli cells, and so the development of mature sperm happened along the seminiferous tubule, along this luminal space here between 2 Sertoli cells. And the Sertoli cells are really providing the nourishment and the support that is needed for these sperm to develop from their primordial spermatogonia into mature spermatozoa. So we start off with spermatogonia, which are kind of the primordial undeveloped version of our sperm. And they must undergo several divisions or several developmental processes. We start with mitosis. So mitosis happens and then we end up with 2 spermatogonia here. One of these spermatogonia will continue to undergo meiosis, which is the development of sex cells or gametes. So only gametes undergo meiosis, whereas all of our cells undergo mitosis. But only one cell proceeds on with meiosis. Another cell stays behind in order to continue dividing to give us more spermatogonia. The one cell that undergoes meiosis 1 eventually gives us 2 secondary spermatocytes, which we can see here. And then each of those secondary spermatocytes undergoes meiosis 2, which is a further division. And then we end up with 4 spermatids. The spermatids are not fully developed. They don't have their tail. They don't have the mitochondria. But when they do acquire those structures, they become fully developed or fully mature spermatozoa. And by this time, they should be at the lumen of the seminiferous tubule. So the development takes place across this space here in between these two Sertoli cells. And by the time the spermatozoa are fully developed, they should be at the lumen and make their way into the lumen of the seminiferous tubule. Now, they're not quite fully functional yet, although they have a tail and the head and the midpiece and all of that. They're not actually fully functioning until they get into the vas deferens. So we'll talk about that as we go on. So spermatogenesis is the process of development from the basement membrane all the way down to the lumen, and then this follows mitosis. So mitosis is basically the division of a spermatogonia. One of those spermatogonia will remain to produce more spermatogonia, but then one of them will go on to undergo meiosis, where it develops into a mature spermatozoa. The mature spermatozoa are released into the lumen of the seminiferous tubule. They remain immotile for a couple of days, about 20 days. And then when they get to the epididymis, and there's the peristalsis, there's the luminal fluid, which is also secreted from those Sertoli cells, then they will acquire their motility. Remember, the fluid has the fructose that's going to be important, energizing these cells. And so they're not quite motile until that 20-day period has passed, and they make their way into the epididymis. From the epididymis, they move into the vas deferens, also by peristaltic contractions or rhythmic contractions, and then they remain in the vas deferens until ejaculation, which is part of the male sexual response. This is controlled by the autonomic nervous system. So the process of arousal, erection, emission, and then ejaculation, all of that constitutes the male sexual response. And this is how we go from sperm that's sitting in the vas deferens to that sperm being released into the female reproductive tract in order to fertilize an oocyte. So there are 3 phases involved here. The erection process is controlled by the parasympathetic nervous system. That's part of the arousal or the initial stimuli for ejaculation. The emission process is where we switch over to the sympathetic nervous system. And emission is really the movement of spermatozoa from the vas deferens into the urethra itself. So the urethra is a part of the tract that leads out to the opening of the penis. And then finally, ejaculation, which is the 3rd phase here, also under the sympathetic influence, is the final release of that fluid. So at this point, it's going to be called semen, when the spermatozoa mix with the secretions of the seminal vesicle and of the prostate, and that is expelled out of the penis as the final part of this sexual response. The stimuli here are going to include the mechanoreceptors of the penis, so they're actual touch receptors on the penis, as well as mechanoreceptors in other erogenous zones of the body. So areas like the neck have certain receptors that are called erogenous zones, other parts of the body as well. And then other stimuli are going to be the higher centers of the brain. So in the absence of physical touch, things like thoughts and memories and visual perception can also descend and have an influence on these phases of the sexual response too. Okay. So here's a bit of a flow chart that describes the first part of that sexual response. So this is basically mechanical stimulation of either the penis or other stimulation coming in from higher centers in the brain will make their way down to the spinal cord. That will increase the parasympathetic influence. Remember, erection is under parasympathetic control. So we want to turn on parasympathetic influence to the penis and turn off sympathetic neurons to the penis. That results in the blood vessels that are moving towards the penis becoming engorged because they relax the smooth muscle. And so blood will fill into the erectile tissue of the penis. That then results in increased blood flow increased blood volume. So once those vessels become engorged, they exert a certain amount of pressure, which compresses the veins of the penis and kind of traps that blood in the erectile tissue. So basically it prevents the blood from flowing out of the penis, which would of course result in the penis becoming flaccid, which is what we do not want in this initial part of the sexual response. So increased blood volume kind of creates pressure on the veins, compresses the veins, traps blood in that erectile tissue and prevents any outflow of blood, which essentially is a positive feedback loop. So more blood is coming into this erectile tissue and is being stopped from leaving. And so that happens over time and produces a full erection. So this is how we go from mechanical stimulation or higher center input from the brain resulting in an erection under the influence of the parasympathetic system. The second phase, is emission, and this is under the influence of the sympathetic nervous system. So here we kind of have a switch from parasympathetic to sympathetic response. This is picking up where we left off from the mechanical stimulation, which resulted in erection, now moving to emission. Emission happens when contractions of the vas deferens, contractions of the epididymis, right, part of the testis, as well as the ejaculatory duct, which communicates to the urethra, all of those structures begin to contract, and we have the movement of sperm, which is spermatozoa, the actual sperm, mature sperm, as well as secretions coming from the seminal vesicles that we talked about and from the prostate, moving into the urethra as well. So the combination of the spermatozoa as well as the secretions from these glands combined, that is what we call semen. So semen is the fluid coming from the glands as well as the actual spermatozoa coming from the vas deferens. And then that can move into the urethra in preparation for the final phase, which is called ejaculation. So in the final phase, again, we're picking up from the mechanical stimulation. This eventually leads to the contractions of smooth muscle in the urethra, but then skeletal muscle throughout other parts of the penis as well. So those contractions eventually expel the semen that is in the urethra out of the body and hopefully into the female reproductive tract. Okay, this is the final phase of the male sexual response. So basically we've got erection, which is parasympathetically controlled, and then emission and ejaculation, which are controlled by the sympathetics.